that's why that control is there. There's still extra microbes in there. There's also extra microbes because this is all done in tanks, right? So you're already up at least twofold of microbes at that point.
Okay, so I just had to do like the hydro with the LV. We had to get over that issue because they would just die within a day.
Exactly, yeah. No, this is still, you still have that problem. That's, in this case, we have a real control for LV, right? So, all right, does that sound good to everybody? Okay. All right, so then putting Davey's stuff together with Linda's stuff, this is basically what Becky did. And in this case, we didn't have the atom system because it's such a pain in the ass to set up. And so what we have here are these tanks out in Hawaii, and a different coral type actually. And they put them in this, and they ran that pump system that Mark made, which is basically a big syringe pump. And so they would have water running across this, and then they would push the stuff into these individual replicant corals. Okay? Then isolate the viral communities and just do the DNA sequencing and on that. Okay? And this is the one you guys know about, like where it looks like what happens is that the nutrients, the temperature, or the pH changes increase the herpes viruses associated with the coral. Okay? And we think that that's probably just because we're stressing them out a lot in this system. And the reason we think that is that it's really quick. So this is where we're treating the corals in the aquarium. And then we did, once we'd seen this, then I sent her back because I didn't believe it. So, and then we did real-time PCRs. Okay, so now we have real-time. So what you can see is that the stressors basically induce the herpes within the first 4 to 8 hours of the thing happening. And so that really says to me that it's something like, that it's basically a herpes that's already living there and that you're inducing it out of the system. Okay. And I think that everything else, so at the time we really thought that's beautiful because of course this is, these are the corals, so they've got the first immune system and they've got the first neural system. And since herpes viruses, we always think of them as neuron associated, which isn't actually really correct, but that's how we think about it. That's where they would be, and I don't think there's any evidence to that anymore. I think that basically what we would guess is that these herpes viruses are actually coronaviruses that are being induced out of the zooxanthellae, if you were guessing with the microscopy data that's available at the moment. Everybody agree with that? Do you agree with that, Steve? Yeah. I mean, there are some pictures that suggest there are herpes-like viruses in the tissue, but even there, they could have easily come from the zooxanthellae and then be in the tissue when they caught them, right? So I'll bet you that that's what's going on. All right, and then this is just the same exact experiment, but what we're going to do in this case is do the microbiomes. Okay. And here, what you find is that all of the treatments increase the number of known pathogens associated with the corals, right? So any of our stressors increase the known pathogens. But this is actually the one that I think is more important. It's not just that by taxonomy they are, it's actually that by virulence factor. So if you look at the metagenome, you see this increase in genes that we know are virulence factors in all 4 treatments. Okay? And probably more importantly is the fact that they do it in different ways. So this is just the same data, but we're going to look at it more specifically. So here are some different virulence factors, and here are some different stress response factors. And what you can see is that there's actually just different patterns of the relative abundances of genes depending on the stressor. Okay? So what that means is that you should be able to tell what disease you're looking at or what stressor you're looking at just by asking what these gene contents are. And we've seen that work in a number of places. So I think that works basically true too. The other thing is you see definitely things where specific microbes are responsible for the pathogenicity signal. And this is the heat treatment right here. And so basically we did not observe any vibrios, and so there were so few vibrios in the healthy coral that we didn't observe them in the Mediterranean. But then when you do the heat stress, suddenly all of the changes in subsystems that we saw were in Vibrios, which meant the Vibrios just went through the roof. Okay? And we've observed Vibrios being important in this system a lot, so it's worth thinking about. Okay, so these are where we are right now. Now the other thing that we've got, okay, so then I'm going to take you through, of course, how we look at it, this healthy reef system, or at the bigger scale. So you guys are all kind of familiar with this, I hope, right? This is the basic, the way right now that we're envisioning the bottom, remember, are these benthic groups competing for space. So you've got the coral, you've got this coral encrustose algae, the CCAs, and then you've got turfs, which are these, basically red algae with cyanobacteria, and they're the little smudge stuff on the reef, okay? And we think these are primary players. I'll show you why. And then we've got the fleshy or macroalgae, so the guys you can actually see. So these guys are all playing there, okay? And then you've got, on a really nice pristine reef, you've got lots of grazing going on, keeping these guys in check, okay? And then shunting the energy very quickly up through the predators to basically the top predators, okay? So this is, and the argument, remember, is dynamical. It matters the rates at which you move this stuff, right? It's not just about standing stock. The other thing that's going on is that if we wipe these guys out, right, we're going to change the grazing rates here, okay? So our model of the holobiont, at least the coral holobiont, is that it's controlling the amount of DOC through the mucus, right? The quality and the type of, and the quantity of DOC that the microbes get, which selects up a specific microbiota. That's why we see that. And then that feeds back, and these guys occupy space on the coral, and that keeps other microbes from coming in and getting it. Well, that would be our guess.
Do you need space and not spice?
Space.
Okay.
All right, and then a fish reef, what happens is that basically the first step, nowadays I'm adjusting how I'm thinking about this, is the turfs become more important really quickly. So if you remove grazing on turfs, they can grow really quickly. And by really quickly, I mean a matter of days. Okay, so they go from almost unobservable to something fairly large. Okay. So that's what happens if you reduce grazing pressure by this clade here. You'll get more turf algae. Okay? And then the turfs, I'm going to call evil for right now, because they're going to be feeding the DOC into the system, which then feeds the microbes, and presumably a different microbial community, which then goes in and causes disease. This is what we call the dam hypothesis, right, for DOC disease, algae, and microbes. Okay? And remember, this is important because this is a positive feedback system, right? So this is making space for more algae. Right? And so you can envision that there's going to be at some point where you're going to have enough algae that the grazers, even if you add a whole bunch of grazers back, are not going to be able to control it anymore. Okay? And that would probably be your tipping point that you're looking for on the coral reef, I'll bet you. All right. The dam, if you put everything together, it basically favors potential pathogens. There's this increase in coral algal interaction zones. I'll show you this. And at least in the first step, it's the turfs that are bad. Yes? Sorry.
So even though you do still have viruses around and you have microbes growing up, but most likely what's happening is the viruses are probably adhering to the mucus.
No, you've still got all those dynamics going. It's just that, the total energy in the system decides where your carrying capacity is, right? It doesn't mean that the microbial food webs and stuff aren't changing, but so it's just that you're feeding more into the microbes, so you're getting more microbes. It doesn't mean that viruses aren't killing and protists aren't killing and all of that other stuff.
Overwhelming.
Yeah.
I'm not sure if you said, but what kind of bugs are on a healthy coral?
Well, that's a good question. I don't remember at all. I think they're alpha, beta, proteobacteria. There's gammas.
Heterotroph? Because the bad guys are the heterotrophs, like Vibrio and Entrix, right?
but it's more like your really super fast growing guys. So it's staph and streps and crap like that, and E. coli and Enterobacter looking things are base, and that's what we see in the Dinsdale stuff, and actually we see that in general. Vibrios seem to be a great indicator species in lots of systems, but in some cases we don't see the Vibrios, we see other things come up. There's also a lot of Bacillus clostridium, and in fact, I didn't show you this, but in the original papers, if you have Diploria strigosa, which has that really thick mucus layer, the diurnal cycle must be really cool because you have a ton of anaerobes living there. So it looks like what you're probably doing is that during the day you're doing lots of aerobic stuff, and then at night you're doing a whole bunch of anaerobic stuff, at least in that one, right? So that's just one of the things that nobody's actually studied much, but I'm sure it looks that way. All right, and then so for you guys in the lab to get more familiar, what we're going to nowadays, we have all of these studies out in the open facility with NOAA, and these are some data taken out of that. And basically, this is Tracy's stuff, right? Well, Tracy didn't do this part, the NOAA people did this part. But what this is that you see basically